Hello and welcome back to Educator.com and welcome back to Biochemistry. So today we're going to continue our discussion of sequencing a peptide or a protein, and we're going to talk about how to sequence larger proteins. And it's exactly what you might think it is. Basically, you just take a larger protein and you chop it up into fragments, and then you deal with those individual fragments, and then you just arrange the fragments. So let's just go ahead and jump on in. Okay. So before we actually get into sequencing the larger peptides and proteins, I want to talk about one other method that's used to identify the N-terminal amino acid. So, because it is going to come up, so another method for IDing the N-terminal amino acid. And again, I'll just use AA for amino acid. Okay. Frederick Sanger who worked out the amino acid sequence of insulin, developed a reagent to identify the N-terminal amino acid. Okay, it is called 1,4-2,4-dinitrobenzene. I tend to separate the words. You'll see it as one word, dinitrobenzene, small b, but I tend to separate. It doesn't really matter. So we'll just refer to it as FDNB, fluorodinitrobenzene. Okay, so here's the procedure. So we'll take our amino acid. Let's just go ahead and go, let's see, H3N plus, we have CC, we have that, and over here, actually, you know what, I'll put the O a little bit better, and over here I'll just write peptide, because again, we're just going to be concerned with the N-terminal amino acid, so this thing right here. And I'll just make it generic, so I'll put an R1. So what we do is we react this with our FDNB, which I'll go ahead and draw the structure. So let me just write FDNB, and the structure is this. 1,4-4-dinitro-benzene. 1-fluoro, 2, 3, 4, 1, 2, 3, 4, 5, 6 numbered carbons. So we react it with FDNB, and what you do is you basically just create a derivative of this. So this is like a label, just like what we did before. You're just labeling the amino acid so you can actually identify it. So what you end up with is with the following. So let's go NCC. I'll write peptide. Then we have a carbonyl here. We have our R1 group. We have our H. And we have our this right here. I'll go ahead and put the NO2 over here. I've just flipped this one around. Basically, you're just replacing this fluorine with the N-terminal amino acid. That's what you're doing. So it just acts as a label. That's it. So once you go ahead and form this, once you've actually labeled this, so let me go ahead and put a little circle around our amino acid. So that's our N-terminal amino acid. And then what you do is you go ahead and react this with 6 molar HCl to hydrolyze, to completely break apart every single bond, just to free up every amino acid. And then when you do that, what you end up with is, of course, the labeled terminal amino acid. You're going to end up with this. You're going to have the R1, you have H, and then of course you have your label. So NO2, and I'll just put the NO2 over here, plus free amino acids. That's it, free amino acids. when you use strong acid like that, 6 molar HCl, you're just going to completely hydrolyze the protein into all of its free amino acids. Okay, and let me write here again. 6 molar HCl breaks all the peptide bonds. Breaks all the peptide bonds. And of course, this thing right here, this is what we're going to be, we just want to identify the N-terminal side. That's it. That's all this Sanger reagent does, is it identifies the N-terminal amino acid. Okay, and now you're probably wondering to yourself, well, why would we even bother with this if we can just go ahead and use the procedure that we used before, the Edmond degradation, and just identify all of them and sequence them that way? You'll see why it's actually important in a minute when we start sequencing larger proteins. Okay, so now let's go ahead and start talking about sequencing larger proteins and peptides. That's fine, I'll go ahead and keep it in black. So, well, it might be nice if I actually wrote down the words properly. How's that? So, sequencing larger peptides and proteins. Okay, as we said in the intro, these have to be broken down. These have to be broken down into smaller fragments. And these fragments are sequenced just like before with the Edmond degradation. Are sequenced as before. So let's say you have a protein or a peptide and you chop it up into 4 fragments. Well, you sequence those 4 and then you have to use, and then you sort of, well, you'll see the procedure in just a minute, but essentially what you do, then you have to decide which fragment is in front of which fragment, and we have some techniques to deal with ordering those. So that's all that's going on. You're just making your job, you're doing the same thing, except you're doing it for more fragments. Okay, not only does the protein or the peptide, you have to break it up into smaller fragments, but you remember these disulfide bonds, the disulfide bridges that exist between the cysteine residues, they're actually covalent. We have to break those because they interfere with the sequence processing. So we also have to break the disulfide bonds in a protein. Also have to break the disulfide bonds. And that's usually the first thing you do. You break the disulfide bonds, then you start chopping up the protein. Okay, now let's talk about the chopping up the protein part, and we'll get back to the disulfide bonds in just a minute. So there are enzymes called proteases which catalyze the breakage of peptide bonds. The breakage of peptide bonds next to specific amino acids. So the fragmentation process, or the actual fragmentation, is predictable and reproducible. And this is important. Predictable and reproducible. So we use enzymes called proteases to actually take that really, really long protein or peptide and chop it up into specific places. And depending on what enzyme we use, it actually cuts the peptide chain next to specific amino acids, maybe 1 amino acid, maybe 2 amino acids, but it does not mess with all of the other bonds, not like the 6 molar HCl does. So these proteases are perfect, and because they cut the peptide chain next to specific amino acids, They're reproducible and they're predictable, so we know exactly where they're going to be cut. And that's what's really nice. Okay, so in addition to enzymes called proteases, we also have some chemical methods that do the same thing. So chemical methods also exist. Chemical methods also exist for breaking peptide bonds at specific points. For breaking peptide bonds next to specific amino acids. Okay, now, as a reminder of what I just said, so nota bene, these enzymes, chemicals, they break single bonds. They break single bonds to create short fragments.